intact and leave the topoisomerase out, reforming the entire DNA. Okay? So this is one way in which the solution for the problem of entanglement is achieved. And this does not require ATP. And that's called topoisomerase 1. Now, topoisomerase 2 uses another mechanism called gating. And this is also intuitive and easy to understand. Here there's a, you can imagine the enzyme looks like this. It has a gap in between, okay? It looks like this. And all that the enzyme does is grabs onto two strands of double-stranded DNA and switches their position with respect to each other. So it makes kind of a gate in between and switches them across. Okay? You can imagine in here, it's as though, let's say, this particular circle is on the outside and the other circle is behind. Then it'll just switch the two with respect to each other at this junction. Okay? That's the kind of reaction that it does, and that requires ATP. So what's shown here is let's say, two different helices, the red and yellow helix. It literally binds these two and switches one across. Because it needs to do that, clearly it needs to break the bonds on both sides of the DNA, right? Both strands of the DNA. Pushes this red helix through, reseals back. That's it. Okay? The crux of the reaction is just pushing one strand, one double helix across another double helix. And it requires ATP. It's called topoisomerase 2. It covalently attaches to both strands. Remember, topoisomerase 1 attached to only one strand and swiveled the other strand. But this one covalently attaches to both because it has to hold on to the two strands until the red helix gets across. Okay, so this is simply an illustration of what I just told you. You only need to know this at the level of detail that I just described to you. That is, making covalent attachments to one helix, pushing the other helix through the gap generated in between, like this. Okay? So that now it's on the other side, and what you have are two different DNA molecules, or two parts of a very large DNA molecule that are now unentangled. So that was the problem of entanglement. The next one we'll spend quite a bit of time on, which is the problem of getting one copy of DNA, no matter what kind of DNA you have and no matter how much of DNA you have. So we have billions of bases in humans. E. coli has on the order of megabases, and yet both organisms ensure that DNA is replicated once and only once in the cell. Okay? So that is a big problem, and how that's achieved is what we'll talk about next. The key to achieving one copy per cell division is having places in the DNA which we could call as origins of replication, and then regulating those origins of replication. You can think of that as sort of the ignition key to go through the whole process of replication. You make sure that it fires only once during each replication. So here's a view of DNA replication from, let's say, some arbitrary starting point in a circular DNA. We call that the replication origin. They have high AP content usually. So you should remember that GC is much more difficult to melt because there are three hydrogen bonds between G and C than AT, which tends to be one of the reasons why replication origins have high AP content. Then you have a local opening of the helix. RNA primer gets synthesized by primase, and you have leading strand synthesis beginning on both strands. Okay? So once the leading strand synthesis begins, now that's a little odd, so think about that just for a moment. In both strands, the first thing that's happening is leading strand synthesis. Okay? What does that mean? That's because everything that we talked so far about DNA replication was from the point of view of one replication port. Okay? If you start over here and go this way, just that one replication fork. So we talked of one leading strand and one lagging strand, right? But what's happening is if you start somewhere in the middle of DNA, you typically have two replication forks going on both sides. Therefore, you have two leading strands and two lagging strands. So step one is the formation of two leading strands, and then you have two lagging strands forming two different replication forks going in both directions. So here's a closer view of what happens. Again, the thing to remember is very simple. You have to begin at the origin of replication. You can imagine the step that's really important first is that unopening. And the way that it happens is you have initiator proteins that bind to this AT-rich origin of replication. The helicase gets loaded already because for the leading strand to even start, you first need to unzip. So the helicase is loaded using a helicase loading protein, and it gets working, opening up this region of the DNA. You have primers, RNA primers that are dropped, and then you have leading strand synthesis that begins. Okay? And the remaining strands proceed in much the same way. Okay. So given that is the closer view of the mechanics of how DNA replication starts, how could it be regulated? That's what we're going to talk about today. So here's another EM view of it. There are two replication forks, and there really are two. You could catch E. coli replicating halfway and look at its DNA on an EM, and it will show you the two different replication forks that exist. So in green schematized here for E. coli would be the origin of replication. We'll talk about how that's regulated to ensure that it fires only once during each cell cycle. So eukaryotes and prokaryotes solve this problem in different ways, and we'll talk about both the ways in which the problem gets solved. In prokaryotes, there is a way to mark newly replicated DNA as being different from old replicated DNA. And that is through the methylation of an A base in a GATC motif, which is shown in green here. Okay? Usually it is kept methylated by an enzyme called DAM methylase, where it's deoxyadenosine methylase, so DAM itself is a methylase. And that keeps the origin fully methylated. Now, when replication happens, the newly made DNA in red here is not methylated. And therefore, this hemimethylated or half methylated state can be seen as different by the rest of the cellular machinery that's in cells. So they know that this is newly replicated, and after the whole round is done, after some time, this becomes competent enough for methylation by the DAM methylase putting on the methylation mark. And now it's ready to fire again. And in prokaryotes, this regulation is simply done by the time it takes to methylate the hemimethylated DNA. Okay? So that's how it's ensured that you get one copy per cell division. Now in eukaryotes, the problem is much more complex. First off, you have way too much DNA to have only one origin of replication. And anyways, you have many different chromosomes, so you must have in each different chromosome one particular origin of replication, right? And you have these cell cycle phases. We'll talk about this in later classes, but I think you're probably familiar with most of this. There's a synthesis phase, which is where DNA is going to get replicated. Then there's a whole bunch of checkpoints that will happen during the G2 phase, or growth 2 phase. Then mitosis, where you have the division of the cells. Then they can remain in this growth phase for varying periods of time. So during all this, you need to make sure that the DNA replication is happening once and only once, right? They have a large genome, they have to have many different origins, and yet it has to be made sure that you get replication only once. So think about that for a moment. That in itself is really much more complicated, because you can't have a simple mechanism where you say, I'm going to use this replication once, I've started, now I'm going to come back, one cycle done, I'm done with the replication, now it's 1 and 1. You have many more replication origins, and they all need to be regulated. So this will be slightly more complicated, so I will give you the overview of what's happening, which is what I'd like you to remember, and then walk you through the individual steps as they happen. So as I mentioned already, there are many replication origins per length of DNA. Here's chromosome 3 from humans, I think, where you have, or yeast, I think, from which you have many different origins of replication. Not only that, it's even more frustratingly complicated because not every origin of replication is equivalent. Some of them will fire or drive replication 90% of the time. Some will fire 10% of the time. You don't need to know which is which or anything. This is just for you to understand that not all origins of replication in eukaryotes fire all the time. Within each of these origins of replication, which are about that big, you could say, that's about 200, right, nucleotides. You have an origin recognition complex binding site. That's what the ORC stands for. We'll come to this in the next slide. Then that A-rich region where you can have unwinding, much like what we discussed for prokaryotes. And then auxiliary protein binding sites where all sorts of other proteins bind. Okay? You don't need to know what these things are that bind, but I want you to remember the origin recognition complex binding site and the unwinding region. You have to recognize the place and then you need to unwind. The unwinding region is just like in prokaryotes, AT-rich. Okay? So how is one ensure? I'll walk you through step by step, but don't look at the slide for a moment. Let's just think about exactly how this is happening. The basic idea is this. You have origins of replication. You fire the origin, and then you make sure that until the cell cycle is done, that same origin doesn't fire again. Okay? So because the cell cycle in eukaryotes can be quite complex, you have regulatory mechanisms that make sure that whatever change you made to the origin of replication, say by phosphorylation, doesn't get removed until the cell cycle is done through, let's say, the action of a phosphate. That's the crux of it. Okay? So here's the origin recognition complex in blue, which is binding the ORC region. And there's a bunch of proteins, you don't need to know what these proteins are, that bind and keep it inactive. Okay? In addition, you also have a helicase in mammal called MCM. That doesn't matter, it's just the replicative helicase that also binds. And this whole assembly is called a pre-replicative complex because you haven't started replication yet. And the two facts that you need to remember here are you assemble all of the proteins at the origin of replication, but it is held inactive until it's phosphorylated by cell division kinases, or CDKs. And those become active only during the S phase to drive the synthesis or drive the firing of origin of replication, or at each origin of replication. So cyclin-dependent kinase or cell division kinase mediates this phosphorylation event. Whole bunch of things happen, you don't have to worry about it, until this origin recognition complex itself is phosphorylated. Okay? So now, rather than marking the DNA, you've marked the protein that binds to the DNA, and that's a state in which you can have replication begin. So helicase goes and everything else happens consequently. Okay? And then, now when you finish, you have a different state here. The DNA is duplicated, the origin recognition complex is bound, but only the phosphorylated form is bound. And that is not permissive for replication. The phosphorylation has to be removed for replication to proceed, and that does not happen until after the cells have divided. Okay? So just to be clear, in eukaryotes you have many different origins of replication. They all fire only once. Not every single one of them has to fire, but anything that fires cannot fire again before the cell divides. And that regulation is maintained through phosphorylation. So when we figure out where the origin of replication is, it's usually done by saying, I'm going to take some random piece of DNA, I'm going to put it inside cells, and I'm going to see if whatever you need assembles there and starts replication from there. The sad truth is we still don't know precisely all the things that are needed for firing DNA replication. And in eukaryotes, the situation is quite complex such that the same piece of DNA can do more than one thing. It can serve as a replication origin, but it can also be something like a promoter that drives the expression of a gene. So here's an example of mammalian origin of replication. As you can see, it's huge compared to that one in yeast, which was only 200 base pairs. This is 50 kb on this scale, so the whole thing is about 100 kb, which is huge. And all that we know is if we delete some little bits of DNA, what happens? So we see that these two different regions, if you delete, you lose the ability to replicate starting from this origin. But this other region, which is at the anterior part, the front part here, also activates transcription of this gene way over here. Okay? So from the next lecture onwards, you will hear about gene regulation, and you'll begin to have an appreciation of how complex that is. And you can have very distant elements affecting genes that are located pretty far away. Okay? So it's pretty complex in humans. That's the message I want you to have. And it's not that one piece of DNA does one thing all the time. The second problem with eukaryotes replicating is that they have histones. First of all, the origins are really complex. Secondly, they have histones, so at the end of replication, the histones also need to be changed. There are two aspects of this that I want you to remember. One is simply getting the histones back, so you double the amount of histones in the DNA as soon as you replicate the DNA, because now you have twice as much DNA. The second thing is getting the post-translational modifications that are present on the histones back again. These post-translational modifications like phosphorylation or acetylation that are present on histones are important for gene regulation. So just because you replicated DNA, you can't forget all the things that have to be done with that DNA. So all the post-translational modifications have to be brought back. So those are two aspects we'll go through one by one. The first one is just simply getting the DNA, getting the histone onto the DNA. The way that's done is by treating the two different oncomers differently. When you have H3, H4 tetramer, that is distributed randomly across both strands, as shown here. But the H2A and H2B are kicked out as replication happens. And then when new histones are brought in, you would need chaperones, proteins that will bind the histone, help structurally to position the histones in the DNA to put back the new histone. So that's shown in this next slide. You do not need to know the names of these chaperones, but you should know that because putting histones in DNA is an energetically unfavorable reaction, you would need chaperones to load histones on them. So histones are loaded into these half-formed regions. You have H2A, H2B loading chaperones, and you have H3, H4 loading chaperones. They can recognize that this is newly replicated because unlike the fully replicated DNA, they will be only partially occupied by histones. Okay. The second thing that I said for histones is you have to now remember all the marks that were there. We'll talk about this in greater detail in gene regulation in later lectures. But for now, all you need to remember is that there are marks. Okay? These blue boxes, if you will. Consider that as a methylation. Then what happens is when these newly synthesized histones are put in, they don't have any methylation marks on them. But let's say the methylation mark signifies you need to express this protein, and it's an important protein for the cell. Every time it divides, if it forgets it, then the cellular function will be impaired. How that's ensured is by having reader-writer proteins. A protein like this that's shown in green, which is able to read the mark by specifically binding, but at the same time able to put on the mark on another newly synthesized histone. It doesn't have to be one protein, it can be a complex of proteins that ensure that this happens. Okay? So at the end of eukaryotic replication, not only do you have 2 copies of DNA, you have histones on both copies of DNA, and you have the marks that were present on the histone re-established. The final problem that we'll talk about for this 1 copy, which is different in eukaryotes from prokaryotes, is what's called the end replication problem. And this really comes from that requirement for RNA primer for the DNA polymerase to start replication. So in blue here is the RNA primer. You can imagine it's going from 5' to 3', replication in red, or in green for the lagging strand. In both cases, after you finish, or here even if you, as you start, there's a gap at the end. So what will happen if the cell keeps dividing and keeps dividing its DNA, progressively you'll get a shrinking from the end, and you could be left with no DNA, depending on the number of cycles that you go through. So that problem needs to be solved, and it's solved very simply by having repetitive sequences at the end and encoding the primer for that in another protein. Okay? So not having any important sequences at the end, and having a mechanism for regenerating the end sequence alone. And that mechanism for regeneration is through an enzyme called telomerase. I think this also won the Nobel Prize a couple years ago, maybe last year. So what the telomerase is a protein complex that has also within it an RNA. The RNA serves as a primer for putting on some sequence, even at the end of DNA. Okay? So that then allows you to keep synthesizing even without putting on an RNA through the DNA primase. So the primase is not required because the telomerase itself has an RNA primer. So here's how that looks. Here's the parental strand, and let's say here's the end where there's a gap. The telomerase has RNA sequence that's complementary. You have the T and the A, the G and the C, which now allows extension, again going in the 5' to 3'. And now this is much longer. Primase can come in and finish it off. So imagine this going on again and again and again and again, so that you have a long stretch of repeated sequences that don't matter too much, except to preserve the rest of the sequence. And here's how that looks. You have a very long stretch of, let's say, telomeric sequence, and that final single-stranded end is kind of tucked in a structure called the T-loop that protects the loose single-stranded ends. So all of this can be simply repeated sequences that don't matter, but now the ends are protected, and every time you replicate, you don't lose DNA. Okay? So that's how the 1 copy is maintained. So not only do you not have more than 1 copy, but you don't lose DNA every time you replicate in the characters. So everything that I've said so far is really applicable for any replication that happens. But you can imagine it's particularly important for the case of germ cells, which are continuous, as I told you in my very first lecture, since life began almost, right? So your parents, germ cells are all ones that gave rise to you. So you really need to make sure that it's kept accurate and transmitted across with very high fidelity, the DNA sequence. So I said already that 1 in 10 to the 5 was the accuracy rate of just 5 to 3 polymerization. There are 2 other steps that lead to increased accuracy. One is a mechanism called proofreading, where if the polymerase made a mistake, it can autocorrect. The second one is called strand-directed mismatch repair, where the cell knows which is the newly synthesized strand, and so it guesses that must be the one that's wrong and fixes it. Okay? Those two processes together give you this final rate of, okay, 1 in 10 to the 5 times 10 to the minus 2 times 10 to the minus 2 gives you 10 to the minus 9. That's how you get the high accuracy of DNA replication in effect. So the first aspect of how the polymerase knows that it's the right base is through structural changes. When a new base comes in, if it is perfectly base paired, the fingers and the thumbs tighten around the base. Okay, that's shown here. If you compare the fingers and thumbs there to there, it's tightened around the base. That gives it the first step of high fidelity. The second different thing is, let's say accidentally some other base came in, that can be excised and repaired. So what's shown here on the right is right at the end you have C instead of A in a stretch of A. Now that's not fully base paired, and that is not compatible with polymerase extension. And this lack of ability to polymerase to extend this is really what ensures that you need an RNA primer in the first place. If you don't have a base paired primer before, you cannot have this extension. Okay? So if there is a mispairing, then there is a mechanism by which the polymerase is able to remove that base, put in the right one, and keep going. Structurally, that's schematized in the next slide, where the base that was wrongly incorporated, or the -1 base, gets removed and a new one gets put in. This site where the removal happens is called the editing site, or the E site. Now this is E and P, which is different from the ribosome exit and polymerization site. This is DNA polymerization, and E for editing. The base is literally flipped out to another region where it's excised, the polymerase steps back and goes forward again. That's the process by which one mismatch at the terminal end can get fixed. The second aspect is strand-directed mismatch, which is where you have a newly synthesized strand, and the cell is able to tell it's newly synthesized because the ligase hasn't yet sealed the nick. So if you have an orange strand and a red strand, let's say, the newly synthesized one may have gaps in it. Okay? And there is a machinery, you don't need to know the name of this, but just remember how it's achieved. It's simply achieved by recognizing that there's one strand that has a nick, and so when there's a mismatch, you don't know which one's right. Whichever strand has a gap, that gets removed and then repaired. Okay? So the bottom line is to first recognize which is the newly synthesized strand. It's newly synthesized because there's a gap, the ligase hasn't sealed it. Remove that and then fix the base pair. The resynthesis will fix the error. The unsealed nick is the clue for which is the new strand. Okay, so all this is fine for that immortal germline lineage of cells, because every time you replicate, you're somehow using this newly made one with respect to the old one and figuring out which one to fix. Okay? And you're fixing that. But what about in the case of somatic cells? So somatic cells refers to all the cells in our body. When we get cancer, when something goes wrong and cells just start dividing like crazy, they have to replicate the DNA like crazy. And they might be triggered by some mutation event that happens. You know, you're exposed to some mutagen. Then how do you fix that? How do you make sure that that doesn't lead to propagation of mutation? Well, there are ways to fix damage in cells. And first off, in this slide is shown the most prominent ways in which damage happens, all the different arrows. You don't need to know all the different changes that can happen, but you'll notice that there are 3 really fat arrows. The fatness of the arrow indicates how common that occurrence is. And all of them are hydrolysis reactions in blue, and they lead to changes in the bases. 2 of them are where the base itself gets removed, which is where the G can be removed from the sugar, and here the C changes. Okay? These two reactions are called depurination and deamination. Depurination because the two bases G and A are purine bases that can then be removed from the DNA backbone, and deamination because the NH2 is changed. So you can see that here. Here's a guanine, which upon hydrolysis, the entire base is excised, or a cytosine where the NH2 upon hydrolysis changes it to uracil. Now uracil is normally found in RNA, but not in DNA. So this would still be a damaged base, right? And you can imagine that once you have such a damage, subsequent base pairing will be messed up, especially changes like this, where you go from C to U. U will base pair with an A, not a G, right? So you have subsequent mutations introduced. And that's shown here. When you have a U instead of a C, in the next round, that U goes on to base pair with an A, whereas the G is fine. So one of those cells now has a mutation that then can never be fixed going forward. So what you need is ways of detecting before replication that something is wrong. Okay? And it's that DNA repair mechanism that's really crucial to allow us to survive in harsh environments. And one of the most important classes of enzymes are DNA glycosylases. These are many different enzymes together referred to as DNA glycosylases because they cleave the glycosyl bond between the base and the sugar, okay, which is indicated here. What they do is they literally recognize there's something wrong, flip the base out, cut it off, and then that gets fixed by the cell. So this class of enzymes are really sort of gatekeepers of mutations. They make sure that any mutation that arises is immediately kicked out. And you can imagine the hydrolysis of each of these bases, A, G, C, T, can lead to many different things, right? So if we just look at deamination type reaction, you can get from adenine, hypoxanthine, xanthine, uracil, thymine. You don't have to worry about any of these structures, but you should know that hydrolysis is the most common way in which bases are getting damaged, and that there are DNA glycosylases that can recognize these damaged bases, kick them out, and then lead to subsequent repair. There's one special case where if you go from a 5-methyl cytosine, which is a base that does occur in humans and many other animals, you can then get thymine. Okay? But this is a normal base that's found in DNA, and it's used for regulation, as we'll discuss later. It's found in a small percentage, 3% in vertebrate DNA. And this is also a normal base. So you're going from a normal base to another normal base, yet the base that's on the other strand does not base pair. So there's something that recognizes the mismatch and then fixes it, as we just discussed a few slides ago. Okay? So the idea here is any kind of mismatch, any kind of base change is recognized by enzymes called glycosylases that are evolved to recognize that mistake. And the first thing that they do is kick the base out. So here's an example of uracil being kicked out. And then there are subsequent repair mechanisms. So we'll talk about those mechanisms in turn. So the first thing that you have is something like a missing tooth, if you will, where the base is missing, and you can think of it as apurinic or apyrimidinic based on which base got kicked out. Now that's then recognized by the cell by an AP endonuclease, or apyrimidinic or purinic endonuclease, and it just removes that sugar off, leaving now DNA polymerase to fill in and ligase to seal the nick. Okay? So key step in base excision repair is you have to excise the base and then remove that missing tooth and seal it with DNA polymerase and ligase. Okay, so it's a very simple procedure. The key thing to remember is that uracil DNA glycosylase, or any DNA glycosylase, recognizes the problematic base, removes that base, that's why base excision, and then you have repair. Now, UV irradiation or any other form of damage can not only change bases, but they can make bases form bonds which are unnatural. Okay? In that case, what can the cell do? There are no glycosylases per se that would recognize this, but this is, for example, a CC dimer that's driven by UV irradiation across two bases that are nearby. Such things are repaired using a mechanism called nucleotide excision repair. Easy way to remember the difference between these two. The first one was base excision repair, where only one base was removed. And as you can imagine, nucleotide excision repair is where a bunch of nucleotides, a stretch of bases, is removed. So if there is a structure that is like a pyrimidine dimer, a CT dimer that forms here, the first one is you have an excision nuclease that lops off a long stretch. Okay? Then the helicase just removes that piece. You have DNA polymerase and ligase. So you should be familiar now that always the fix is the same. You first remove the problematic region. Fix is always you have polymerase and ligase. Okay? Polymerase and ligase will fix the problem. So all of that is for having one side broken. What if you had both sides damaged? Both strands damaged. Then how can you use homology for repair? Well, for that you need some other strand that is also present, or some other DNA copy that's also present, which you can use as something that shares homology to then repair. Okay? And that doesn't happen all the time. So you consider these two cells. Let's say on the left, you have a cell that has both strands broken, but there is nothing that's homologous that's available for it, okay? Because it's not newly replicated. On the other side, you have newly replicated DNA that's available, which is exactly the same sequence. These two get processed in different ways. If you don't have anything that is precisely homologous for you to copy from, the only way is to say, okay, I'm going to lop off some sequences, seal it, and hope nothing bad happens. So the cell can do this called non-homologous end-joining, because not using any homologous sequences, and that can happen at any time. The other option is to use homologous region to copy from that, and therefore fix the newly synthesized or the damaged DNA alone. And that can happen after replication, where you have two copies, but before cell division, where it's not partitioned off into two. Okay? We'll talk in greater detail about homologous recombination later. Okay? So to finish off, I just want to put this table up to remind you that this process of DNA repair and DNA replication is absolutely crucial to keep us from getting cancer. Many of the proteins that are found to be mutated in cancer are proteins that regulate all these mismatch repair pathways that we talked about, or replication. So, for example, mismatch and nucleotide excision-based repair are common in colon cancer, skin cancer, and so on. These proteins get mutated. So, to sum up everything that I've told you today, we discussed the problem of DNA replication from the context of each of these different aspects that the cell solves. You should know how this happens. There may be a question asking you, say, name 3 of these, or solve 1 or 2 of those. And we then talked about how a accuracy of replication and then even fixing mutations is an absolutely crucial part of life, and that any of those mechanisms, if they are broken, you could have cancer. So particularly remember DNA glycosylases and how they are crucial for the different kinds of repair and when those kinds of repair happen. And with that, we'll end for today. I'll remind you again, I won't be here next Thursday. Dr. Fowler will take over next Thursday. This Thursday. I'll be back next Tuesday.